In this lesson, we'll continue our discussion of muscle tissue by taking a look at how the nervous system interacts with the muscle fiber to produce muscle contraction. So we're going to start off looking at the anatomy of the neuromuscular junction. We'll also take a look at the sequence of events that take place there. Then, once the muscle fiber has been excited, we're going to now couple that to an actual contraction, right? So we call that excitation contraction coupling. So way back in our very first lesson, we had said that one of the properties of muscle tissue is that it is excitable, right? It means that it will respond to some type of stimulus. And one of the reasons it's excitable is it has the ability to conduct electrical impulses that we call action potentials. Now, the two types of cells in the body that can conduct these action potentials are going to be the skeletal muscle fiber, which we're going to go over today, and then also the neuron, which is the nerve cell. We're going to see that in this lesson also. Right? So these are the two cells in the body that can conduct electrical impulses that we call action potentials. Now, this is a little bit advanced. We're going to cover this in more detail when we do the nervous system. The two phases of an action potential, right, a nerve impulse, is going to be depolarization. That's going to be like step number one. And then repolarization, that's step number two. I'm not too concerned about this right now in this lesson. I just want to introduce these terms. My last comment here is this: a skeletal muscle fiber will lay around all day long. It will not contract until it is stimulated by the nervous system. The nerve cell that actually innervates a skeletal muscle fiber to contract, we're going to call a motor neuron. Again, neuron just means nerve cell, and then the fact that it's a motor neuron, it means that it's going to be stimulating a muscle. Don't write this down, but that would be in comparison to a sensory neuron, which senses things. This is actually going to cause a muscle contraction. So let's take a look at this neuromuscular junction. Neuro means nervous system. Obviously, muscular means the muscle system. So this is going to be the connection between the nerve cell and the muscle fiber. A lot of times we just abbreviate it the NMJ. Right? It's the synapse, and synapse just means a meeting point between the neuron and the skeletal muscle fiber. So let me show you what's this picture here in your book. This is a little bit advanced. We haven't done this yet, but this right here is a this right here is a nerve cell. All of this is a nerve cell like this, and you can see it's going to the muscle fiber right here. This is the muscle cell. So this would be like the cell body of the neuron, and then this blue tube here is going to be, see that blue tube in there? This is going to be the axon, and then you can see the axon actually fans out so that it can stimulate multiple areas. This is called the, again, a little bit advanced. We're going to cover this in the nervous system. This is called the telodendria. This is just the branching out of the axon. You don't have to know that for now. And for today, just ignore these yellow coverings. This is what we call the myelin sheath. It's on top of the axon. Again, we're going to cover that in the nervous system. All right, so what do we have here is a motor neuron that's sending the nerve impulse down the axon to the muscle fiber. How do I know that? See that little red squiggly line right there? That's showing the nerve impulse or the action potential and what direction it's going to. Now, what they did here, they take a piece of the axon that's kind of going over this way, and they blew it up. So this is the axon, and we're kind of looking into the axon, and you can see the axon is up against the muscle fiber right here. So this would be the actual neuromuscular junction, right? Neuromuscular junction. And what we're going to do is we're going to blow this up now, this section right here, and take a look at the sequence of events. So I have everything written down on the next two slides, the sequence of events. Instead of me going through this line by line, let's do it by going over the picture first and then going back to the notes to make sure everybody understands everything. So I'm going to skip these 2 slides. Again, I'm going to go back to this picture here. This is what we're going to be blowing up now. And I want to give you the normal anatomy first. This is an overview slide from your textbook. I'm actually going to add a little bit of detail. Again, that's going to make our life a little bit easier when we do the nervous system. So we are going to add to this a little bit. But this is actually showing the sequence of events, step 1 to step 2 to step 3, and so on and so forth. But I want to go through each one of these pictures individually. So here's what I did. I actually labeled this. You won't have this in your textbook. You can use it here from the PowerPoint. This is the normal anatomy of the neuromuscular junction. So again, what are we looking at? All of the stuff in blue is the neuron. The end of the neuron, where the axon ends, is known as the axon terminal. Right? So all of this down here is the axon terminal. Within the axon terminal, we have, it's almost like water balloons. These round structures right here, all of these round structures, these are known as synaptic vesicles, right? Synaptic vesicles. But instead of being a water balloon containing water, these red dots here represent a chemical which is a neurotransmitter, right? One of the ones you may have ever heard of is known as acetylcholine. This is the main neurotransmitter at the neuromuscular junction. You may have heard of other neurotransmitters. Maybe you've heard of chemicals like dopamine, serotonin, epinephrine. These are other examples of neurotransmitters. They just don't exist at this level, right? They don't exist at the neuromuscular junction. Now, notice, guys, that the axon terminal here, this is the cell membrane of the axon terminal. Notice that the axon terminal doesn't touch the cell membrane of the muscle fiber. There's actually a gap. This space is known as the synaptic cleft, or you can call it the synaptic gap. Right? So that's an important thing to know, that the axon does not touch, right? Does not physically touch the muscle fiber. There's a space there. We're going to ignore these crescent-shaped structures right now. I'll talk about them later. On this side here, this kind of tan or pinkish membrane, this is the cell membrane. Here it is again, right? The cell membrane of the muscle fiber, right? And here I labeled it. We give this a name, however. It's called the motor end plate, and all that is, it's the cell membrane of the muscle fiber, right? We know the cell membrane of the muscle fiber is called the sarcolemma. It's just the sarcolemma at the neuromuscular junction they call the motor end plate. The other thing I just want to point out, since it's here, notice that the motor end plate actually folds like that, and we see it over here. This is just known as a junctional fold, right? For us today, it's not all that important. I just want to point it out because it is there. So I do want you to know this is the normal anatomy of the neuromuscular junction. Let's take a look now at the steps that take place. So the first thing that happens is, here's that, again, that little symbol showing that the electrical impulse. Let's just say the electrical impulse comes down and it gets to the end of the axon. So this red right here represents the action potential coming down to the end of the axon. All right, so we have the action potential here, but here's our problem. We want to restart that action potential on this membrane so that the muscle fiber contracts. But the problem is we have a gap. Okay, so we have to overcome that. So the first thing that happens as this electrical impulse comes down this way, calcium ions, which are in here, diffuse into the axon terminal. Once the calcium ions come into here, that stimulates the vesicles, the synaptic vesicles, to release their neurotransmitter into the synaptic cleft. So here's my next slide. So notice the calcium went in here, right? I'm not showing that, I'm just telling you that's what happened. The synaptic vesicles then opened up and they released their acetylcholine, in this case, into the synaptic cleft. That process of releasing the acetylcholine into the synaptic cleft, you're going to call exocytosis, right? We release something from the cell. Now, the neurotransmitter once it's released, it's going to be in higher concentration here and in lower concentration here. So what it does is it diffuses from an area of high concentration down to an area of low concentration, right? So all of our acetylcholine is going to migrate or diffuse towards this membrane over here. Actually, let me go back a second. One of the things I forgot to define for you, I'm just realizing it now. In the cell membrane here, the motor end plate, we have these purple structures. These are receptors. They're going to receive the acetylcholine. So I just labeled this as an ACH receptor. It's going to receive the acetylcholine. So notice as the acetylcholine migrates down, it's going to head towards these acetylcholine receptors. Watch what happens. So now we can see the acetylcholine come down and it attaches to the receptor. It's kind of like lock and key. It's a specific type of fit. Once that acetylcholine binds to the receptor, notice the receptor is attached to a channel now that's open. Before, if I go back a minute, see how the channel was closed? But now when the acetylcholine binds to the receptor, a channel opens and sodium will move into the muscle cell. That movement of sodium into the muscle cell is the first part of its action potential known as depolarization. Once the sodium has moved in, and I added this is not in your book, potassium moves out. It actually moves out through its own channel, but I just wanted to show you. Potassium will then move out. That's going to be the second phase of the nerve impulse called repolarization. Again, that's a little bit advanced. We're going to cover that more in the nervous system. But here's the concept, guys. Once the sodium moves in and the potassium moves out, look what we generated. Now this membrane is red. That tells us we now have the nerve impulse on the muscle. This is the action potential on the muscle. And this is now going to travel along the sarcolemma down the muscle. So let's just say the neuron has stopped firing. We don't want this muscle to contract anymore. What we need to do is get rid of any acetylcholine that's in the synapse. As long as acetylcholine is in the synapse, it's going to stimulate these receptors to move sodium and potassium. So we got to get rid of that acetylcholine. So these crescent-shaped structures here represent enzymes that actually degrade the acetylcholine. The name of this enzyme is called acetylcholine esterase. A lot of times we just say cholinesterase. Okay, so this enzyme, this yellow crescent-shaped structure, will break down any of the remaining acetylcholine. And now you can see the action potential is traveling, along the muscle to stimulate the muscle to contract. Okay, let's go back a moment. Actually, I'm going to go forward one more time. So here now we saw that the action potential, remember before the red arrow was over here? Now look at the red arrow is over here, showing that the sarcolemma is transmitting the action potential down the muscle. Right? And we're going to look at what happens next in a minute. So let's just go back over the notes just to make sure everybody has the sequence of events right here. So the first thing that happens is the AP, which is the action potential, is going to arrive at the axon terminal. That's the end of the axon. The next thing I want you to know is that calcium is going to diffuse into the axon terminal. That's important because that calcium then causes the synaptic vesicles to fuse with the cell membrane of the axon and then dump out the neurotransmitter by this process known as exocytosis. And the neurotransmitter goes into the synaptic cleft. From that point, the neurotransmitter, which is the acetylcholine, is going to diffuse across the synaptic cleft towards the motor end plate, right? So that process of going from an area of high concentration to an area of low concentration is diffusion. Once the acetylcholine is across the synapse, it's going to bind to a receptor. Right, so that's going to bind to a receptor. The neurotransmitter binds to a receptor that's going to open up a sodium channel. If you remember, Na is sodium, and then sodium is going to move into the cell. Right, that was that arrow that we drew with the sodium coming into the cell, and that causes the first part of an action potential that we call depolarization. Then, after the sodium has moved in, potassium is going to go the other way. It's going to move out of the out of the muscle cell. And that movement of potassium out of the muscle cell is called repolarization. Repolarization. Once we're done and we don't need any more muscle contraction, the enzyme acetylcholinesterase is going to break down the acetylcholine, right? Any excess acetylcholine that's in the synapse we want to remove. Right, so that's going to be removed. All right, so what we went through again, just to show you, the action potential traveled down to here. Then we saw the sequence of events. Right, the first part was here. Calcium moves in. That stimulates exocytosis. The acetylcholine goes from an area of high concentration to an area of low concentration. It binds to the receptor. So here's this acetylcholine binding to the receptor. That opens up sodium gated channels, right? Sodium moves in first. That causes depolarization. Then potassium goes out second, and that causes repolarization. Right? So as long as acetylcholine is here binding to the receptor, this muscle is going to contract. This muscle fiber is going to contract. When we're done, however, notice the gates are closed again. Right? No, there's no more depolarization or repolarization. We want to get rid of any excess acetylcholine. This enzyme is the acetylcholine esterase. Again, like I said before, I think you can just call it colon esterase. A lot of times in medicine, they just call it colon esterase. And now we have the electrical impulse on the muscle fiber, right? We moved it from the neuron onto the muscle fiber, and now it's going to travel down the muscle fiber. Okay, so the last thing I want to go over with you in this lesson is this. Now that we know how the neuromuscular junction works and how we get the action potential going down the muscle fiber, let's couple that. Let's take the electrical impulse and turn it into a contraction. So what we're talking about now is excitation, contraction, coupling. Again, I'm going to do the same thing, guys. I'm going to skip the notes here for a moment and let's go to a picture. So I'm going to blow this up in a second. What our book is showing us here is what we just went over, the neural control of the action potential coming down the axon. Again, our muscles don't contract unless the neuron tells it to. Then we're seeing the excitation. This is the steps we just went over where the acetylcholine is released onto the receptors of the motor end plate. That's going to be the excitation. So now we want to follow it. What happens next? Once that muscle fiber has been excited, what happens? So I'm going to take this picture now and I'm going to blow this up to make it a little bit bigger. So let's just say this red line here represents the action potential traveling along the sarcolemma. What we need to do is to get the action potential into the middle of the muscle fiber where the sarcomeres are. So if you remember from an earlier lesson, the electrical impulse will travel along the sarcolemma and then go down the T-tubule. This was known as the transverse tubule. This was the structure that brings the action potential into the, or deep into the muscle fiber. Just to review some anatomy. So next to the T-tubule here, this big structure is the Sarcoplasmic Reticulum, and we know that it stores calcium. These green circles here, these balls are known as, or they're going to represent calcium, and notice they're stored in the SR. We also saw that there was a swelling of the SR on either side of the T-tubule. This swelling was known as the terminal cisternae. And remember that you had one swelling on one side another swelling on the other side, and then T-tubule in between. And we call this area a triad. I think the analogy I used was like an Oreo cookie, right? This was like one piece of the cookie here, another piece of the cookie here, and then the cream in the middle was this. Right? This relationship is important. So here's what happens. As the action potential goes down the sarcolemma here, it drops into the T-tubule. This electrical signal in the T-tubule now stimulates the sarcoplasmic reticulum to release the calcium, and the calcium floods the sarcomere. Remember, this is the thick filament, the red is the thin filament, right? And these little things sticking up are the myosin heads, and they face the thin filament. So I want you to picture this for a minute. Let's pretend this was like a tractor trailer truck. We're looking at the tractor trailer truck from the side, right? The side view. This whole red and blue and red over here is, think of it as a side view. But now, because this is a little bit weird, we're looking at this, here they cut it here. We're looking at this filament here, guys, this blue filament right here. This is this blue filament, but now we're looking at the tractor trailer truck from behind, like we're looking at it from where you're driving behind it, right? So that's what we're seeing here. So that's this top one here. Then in the middle, the red line is the actin, also like we're looking at the back end of the actin. And then this blue line over here is this one, right? So we're looking at this tractor trailer truck also from behind. So it's a different perspective where we're looking at the proteins from behind, not a side view like this. So notice when the muscle fiber is relaxed, the actin, its active site, the yellow there, is covered by tropomyosin. This little kind of green ball there represents tropomyosin. The kind of aqua color is the tropomyosin right there. It's covering the active site, and the myosin head right here cannot bind. Right, so this is what it looks like when a muscle's in a relaxed state. So what happens? Once the calcium is released, it's going to bind to troponin. The troponin changes its shape and it pulls the tropomyosin away from the active site. I had covered that in a previous lesson. Right? The troponin physically changes its shape, pulling the tropomyosin away, and watch what happens. The active site is now exposed. So this active site is exposed to the thick filament above, or the myosin above, right? And then this right here is exposed to the myosin below. Once this active site is exposed, watch what happens. The myosin heads are energized. I'm going to go over that in the next lesson, how they become energized. They're energized, they jump up, and they bind to the active site, right? So this fiber here, this blue fiber here, is binding to one of the proteins on the red strand here, and then this blue fiber is jumping up and binding to one of the red strands over here. This connection is actually called a cross bridge. Once the myosin head connects to the active site, we call that a cross bridge. But this cross bridge never would have happened if we didn't release the calcium, right? So that's why calcium is so important to muscle contraction. All right, so I'll go over one more time. We're in a relaxed state, right? The myosin and the actin are not touching one another. Once the electrical impulse travels down the sarcolemma, it drops down the T-tubule. The T-tubule we know is connected to the SR on either side. This electrical signal stimulates the release of calcium from the SR. The calcium comes down and it floods the sarcoplasm. I'm sorry, the sarcomere. I apologize. The sarcomere, right? But it's technically in the sarcoplasm, but it's flooding the sarcomere. Now, the calcium is going to bind to troponin, which is one of the regulatory proteins on actin. Once the calcium binds to troponin, this is in the relaxed state where the troponin and the tropomyosin are covering the active site. Once the calcium binds to the troponin, it's important that you know the troponin physically changes its shape, pulling the tropomyosin away from the active site. So now our active site is exposed. That's good. Once the active site is exposed, the myosin head, these little blue extensions here, they jump up and they attach to the active site. That formation of the myosin head to the active site is called a cross bridge. And in the next lesson, I'll show you what happens from there. The myosin head is actually going to swivel. It's going to actually pivot and now pull the actin. Let's say over here, this is the actin that we're talking about. Pull it towards the center of the sarcomere, shortening the sarcomere. So let's go back over the notes for a second. Okay. So here everything is written. The action potential travels along the sarcolemma, and then it has to go down the T-tubules, down to where the triad is. Once that electrical signal gets down the T-tubule, the next important thing you need to know is calcium is released from the terminal cisternae of the sarcoplasmic reticulum. That calcium floods the sarcomere and it binds to troponin. We said before that troponin is going to then change its shape and it pulls the tropomyosin away from the active site and we expose the active site. At that point, the myosin head is going to jump up and we're going to then start the contraction cycle, which again I'm going to talk about in the next lesson. Okay, so and then this is kind of what it's going to lead to. So once the sarcomeres contract, we'll see that the muscle fiber will contract, and then all the muscle fibers in the muscle will cause the muscle to produce tension. Again, we're going to go over that in the next couple lessons. Okay, so that concludes this lesson. Again, a very important lesson on the neuromuscular junction, and then how we couple that electrical stimulation to an actual contraction.